Thank you. And as Jenny said, I'm Joe, and my topic is the characterization of the skin microbiome of free-ranging common bottlenose dolphins in Alabama waters using a novel technique. So the microbiome is all the microorganisms living on and or in an animal. They're critical for roles in health. They've been seen in respiratory systems to linked with diseases and lung diseases and infections. They're also in the gut, create amino acids and proteins. And it's really important to understand a baseline for health moving forward. And then as you see here in this image, this is the skin, and here's the microbial community that's actually on it and living on it. This is really important for skin defense against pathogens. So specifically for cetaceans, they're difficult to sample because they're cryptic species. They move around fast, they're surrounded by water, and these waters are always changing. And we know that these waters can influence the skin. As you can see here, we have freshwater lesions. That is when an animal is in fresher water than it's used to, and that can cause incisions and lesions, and then that can change the microbiome and can potentially allow pathogens in. And here we actually have some images of a microbial community on humpback whale skin, and the arrows are pointing at unique microbiomes. Specific for dolphin skin, there's 6 core phyla, and studies have shown that skin and water differ between the microbial community composition. And this is still important to take both measurements of, because there are some studies that think that the environmental community can influence the host community. And between coastal and offshore dolphins, there have been studies that show that there's difference. So hosts are significantly different sometimes, and especially with species characterization, they're often different. And no study has actually characterized the skin microbiome of dolphins in Alabama. And before I go on to that, you can see here in this figure that the skin and water have relative abundances, and they actually, you can see that they're different, even though they do have some same classes of microbiome. And why it's important that this hasn't been done in Alabama is Alabama has some very unique waters. Mobile Bay is the 6th largest freshwater discharge in North America. Perdido Bay has similar reactions to Mobile Bay, but it's not as drastic. So why this is important is you see in these graphs over this graph here, you have the mean from 1976 to 2014 of discharge with this red line. And even within that, you have some major extremes in Mobile Bay. And this creates high environmental variability. And as you move further out to the Gulf, you have less environmental variability, which creates a gradient within these bays, which really is important to testing multiple environmental stressors. So some environmental influences that have been shown is salinity has been a major driver in microbial communities in sediment and water. And for this study, they actually took sediment from fresh water and put it into a saline environment, and they put salt water into a freshwater environment. And you can see how the microbial communities have changed, but no studies have examined how this salinity changes impact skin microbial communities in cetaceans. Some methods for collecting the samples for microbiome have been through health assessments and biopsy samples. Here you can see an image of a health assessment where it requires capture of the live animal. This is very labor intensive and expensive. You can see here there's 8 people working on one dolphin, and there's several different vessels as well for support. And then for tissue sampling from biopsy samples, you have a really small sample, and there haven't actually been studies that have looked at swabbing of the biopsy, because for health assessment, what they do is they swab the animal and they can get a much better sample size, because in different locations of the skin, where biopsy is very pinpoint location. For methods, specifically for cetacean skin, it's been either flash frozen in liquid nitrogen and stored until it was stored until minus 80 degrees Celsius, or an RNA later, and then eventually stored in minus 80 degrees Celsius. And you can see here that RNA later has impacted in canine fecal samples that the DNA concentration is much lower than without using buffer. In this study, I also looked at ethanol, but there was one study that compared ethanol and no buffer specifically in skin, but for humans, and it showed that ethanol can also change the microbial organisms that are there. So no studies have compared these methods specifically for RNA later versus no buffer in cetacean skin. The objectives of my project are to determine if swabs of skin collected via remote biopsy yield sufficient microbial DNA for 16S metagenomic analysis, examine how the free-living water microbial community influences the skin microbiome, and if it influences it, analyze how skin microbial community composition shifts in response to changing environmental attributes. We're looking at salinity, oxygen, and temperature and discharge for this. And these three topics will be the highlights for my first chapter. And for my second chapter, we're going to be looking at storage methods. So we're going to be determining if skin swab storage conditions in RNA later versus cold storage affect DNA concentration and microbial community structure. Our hypotheses for these objectives are that the swabs of bottlenose dolphin skin collected via remote biopsy surveys will use sufficient concentrations of DNA for 16S metagenomic analysis. Water samples and dolphin skin samples collected at the same time and location will have significantly different microbial communities, suggesting that they are not necessarily linked. And for environmental variations, for this hypothesis, specifically for salinity, we're going to be looking at how lower salinities will have skin microbial communities with higher relative abundance to a freshwater tolerant bacteria. For oxygen, we expect that in low oxygenation areas, there are going to be more anaerobic bacteria present, and we don't expect there to be much variation within temperature. For the storage, bottlenose dolphin skin swabs stored in RNA later will have lower DNA concentrations compared to samples that were flash frozen in liquid nitrogen prior to storing at minus 80 degrees Celsius. The methods for this project were taking biopsy samples, and this was heavily permitted because it is invasive. We are taking a puncture of the animal, as you can see in this here. Why this is helpful is within health assessments. They're also very invasive, but you don't have to take the animal out of the water. This is real quick. With health assessments, you have the animal out of the water and people on the water for up to an hour. And with biopsies, though, I talked about how small they are. There's an image, they're about the size of the tip of your pinky. And it's taken with a dart with a 10 by 25 millimeter or 10 by 30 millimeter dart. So really what you're doing is swabbing a circle with a diameter of 10 millimeters. As you can see in this image here. So then the skin was swabbed and stored either in RNA or liquid nitrogen before it was frozen in minus 80. The RNA later was used for the mobile base surveys, and liquid nitrogen methods were used for the Perdido Bay methods. For Gulf of Mexico, that's where we're looking at our storage methods. So, and we're doing that because there's less variation. And for that, we started off with 3 liquid nitrogen samples and then rotated to 3 RNA samples and continued that rotation for the rest of the project until the rest of the samples were collected. And with each biopsy sample, we collected 2 liters of water. If the dolphins were in the same sighting, we only collected 1 or 2 liters for them, and they could be potentially shared for that sample. And those were about 500 milliliters of water were filtered through a 0.2 micron filter. And at each location, we also took YSI data where we looked at salinity, temperature, and dissolved oxygen to measure oxygenation in that area. So here we have our survey areas in Mobile Bay. The red dots are where we had biopsy locations. And in Perdido Bay, the red dots are where we had biopsies. And this blue dot was where we only took water sample. And this was because that sample didn't have enough skin to actually swab, but we still wanted to take water there. And when there were shared water, they are highlighted in the same color. For Mobile Bay, we had 40 swab samples, skin swab samples, and 37 water filter samples. And for Perdido Bay, we had 44 swab samples and 36 water filter samples. For the Gulf of Mexico, this box highlights our survey area. So we were only taking animals from that location and sampling animals from that location. The blue dots were the samples for RNA later, and the red dots were the samples that were flash frozen in liquid nitrogen. And again, if they shared a water sample, they were haloed in the same color. For Gulf of Mexico, our survey was ended early because of a hurricane. So we were only able to get 15 swabs. We ended up with 9 that were flash frozen in liquid nitrogen and 6 that were stored in RNA later. And then we had 13 water filter samples. So for actually sample processing, we did DNA extractions using DNase blood and tissue kits. And then we decided to use 16S metagenomic sequencing with the aluminum platform. And what this does is help eliminate any eukaryotic cells. So we don't look at the host DNA. It's looking specifically for bacteria. And we're looking at all the bacteria that are present there, which is what the metagenomic sequencing does. For analysis, to verify the presence of DNA on the skin, we did PCR amplification. To determine the microbial community composition, we're using the program Mother to run bioinformatics. And then to examine the effect of environmental variation, we're using a NOVA test to compare the salinities to the microbiome, and we're using a Bray-Curtis dissimilarity index to compare the microbiome between samples. The environmental variations that we're looking at, again, are the salinity, temperature, and dissolved oxygen. Discharge was calculated by adding the mean daily average discharge for the Alabama and Tombigbee rivers for Mobile Bay, and for Perdido Bay, it was using the Perdido and Styx rivers. And for storage methods, because we're just doing the comparison between RNA later versus frozen and flash frozen and liquid nitrogen, to compare those, we're just using a pairwise t-test. The preliminary results, as you can see by this gel, we were able to extract and amplify DNA, which is great because it means that I can actually study this. So we did a test run where we did 10 skin swab samples and 9 water filter samples from Mobile Bay. And as you can see, the DNA 16S band was amplified. So the implications for this project is that we're establishing a novel method for collecting free-ranging cetacean skin microbiome. And why this is important is it allows us to use more samples from a biopsy. That little biopsy that we collect, that's the size of your pinky, for our project, we collect it for genetics, stable isotopes, contaminants, hormones, and microbiome. So we get a lot out of those samples. And that's important with looking at the free ranging, because you can get a lot out of that little tiny bit and still not be as invasive. This is also going to demonstrate the potential shift with changing environmental conditions. This will allow us to do population estimates to make sure that those animals in the population are healthy in a much better way than, again, having to do your traditional health assessment, which is very invasive and time-consuming and expensive. And it will also provide information on how storage conditions may impact sample analysis, which is important for future. So we know that we're doing the best methods and make sure that we're getting the best results possible in our studies. I would like to thank my committee members, Drs. Carmichael, Bloodgood, Kilrees, Subramaniam, and Walczyk from UF. And then I'd like to thank all of the DISL members that have helped me along the way and our collaborators at NOAA, and recognize that this was funded by Deepwater Horizon Natural Resource Damage Settlement Funds provided by the Alabama Trustee Implementation Group with funding support and participation of ADCNR and the Food Drug Administration. Thank you. Do you have any questions? Yes.
So arguably some of the best biofouling organisms are eukaryotes, and they're microbial in size. So why are you only focusing? Well, what's justification for only looking at 16S? Are you doing any insight by looking at 18S or?
The reason why we didn't want to use the 18S was because then we would get the host DNA. We wanted to rule that out, and we're looking specifically at just the bacteria, the 16S bacteria that are on the skin.
I mean, this is probably coming from a place of ignorance, but can't you tell what the dolphin is versus what other kind of eukaryotic protists are? You know, in the informatics.
Yes, you should be able to tell that. And then you could weed that out through the bioinformatics analysis. It just creates more of a streamlined method.
Do you have, and were you able to keep track of individuals? Were you able to recognize some individuals that you took samples from and know their previous migratory history? Like some dolphins are highly migratory, and so you have some individuals that would stay in that location and some that.
Yeah, so with these dolphins, we actually do photo ID as well. And with the biopsy samples, we take identification shots. So if you go back to this photo, we could also use this fin to figure out which animal it is. And these animals actually don't move too much. They're not super migratory based on what we've been doing with the photo ID. So they like to stay in the areas that they're in.
Because one of the things I think would be interesting is if you know that some individuals are more migratory than others, to compare those more migratory individual samples to those stationary samples and compare how much they differ from the local water quality conditions, since you do seem to have some of that data.
Yeah, that would be very interesting. Yes.
Kind of to Ben's point, is it obvious when you sample? I mean, if they are kind of, you know, they don't move around so much, can you tell like, oh, we've already sampled that guy? Or
so
I mean, because I would imagine you're not going to know all of the fin. I think you do photo analysis for the
right. So a lot of the times it gets very difficult, especially in Perdido Bay, because the population is as big, and sometimes we do get resampled animals. We do our best not to do that, and oftentimes it comes down to, I just saw a hole, don't shoot, hold your fire. Because it's sometimes they're popping up for a little fast bit, and a lot of times we'll be on a dolphin group for 10 minutes, and right when you're about to finally get that shot was the first time that you got a really good look and you see the hole, and then you have to call it off.
So have we shot the same animal twice and compared them?
Yes. We have not done that analysis yet, but there are potentially 4 opportunities that I could do that too.
Yes, we would never purpose.
Yes.
Yeah, so we could. Yeah.
If you need an N of 10, you have to just.
Yeah, there's been a lot of people that have asked, like, can you get a spot with freshwater lesion and a spot without it? And one, you can't, it's unethical to shoot the dolphin twice. And then some people have asked, can you split it? So you have part of it with freshwater lesion, it's not, and it's not that easy to shoot a dolphin. Yes.
Along those lines, aside from shooting an animal twice, have you taken a biopsy sample of a dolphin that was in for health assessment so that you can compare a bigger swab versus the biopsy?
We actually have an REU student coming this summer, and part of their work will be looking at health assessment, and they're going to be looking at the microbiome from those animals. And I could potentially bring that into my.
Yes, so we'll be comparing an actual swab to a biopsy swab to make sure they're show that you repeat for the online audience, what is being said in person?
Yeah, so they just asked what, with health assessment, could I compare health assessment to the swabs? And we have an RU student coming in that's going to be looking at that, particularly this summer.
So I'm sort of curious how many locals have called you in for shooting dolphins?
Can you repeat that?
So he was.
And have they asked for how to do it?
He was asking if people have called us and have reported us for shooting at dolphins. We have gotten those calls before, but before we do these surveys, we alert local authorities that we're doing this, that we have permits for this. It's not like we're just going out and shooting dolphins. This is heavily permitted. We have a lot of restrictions that we have to follow. The boat has a big NOAA emblem on it, so it's very well marked that what we're doing.
I have a question from online. Has anyone cultured bacteria from coastal or inshore dolphins versus offshore and tested the tolerances of the bacteria to salinity, oxygen, etc.?
The question was for looking at coastal and offshore, looking at salinity. There haven't been studies that have actually looked specifically at the environmental variation before in the dolphin skin. Yes.
So what do you think the turnover time is for a microbiome? Like the dolphin gets this huge plume of fresh water. How many days or weeks would it take for that?
So
microbiome?
in other studies looking at the sediment, it was about a month where they saw significant results or after it. There hasn't been any specific studies with dolphins, and that can be interesting because dolphins have high turnover rate with skin. So seeing how the microbiome changes with that would also be interesting to look at.
So it looks like you changed your storage between your locations and between these sampling years. So what made you change the way you were storing your swabs? And are you worried that your results from your second chapter are going to sort of indicate that one of your groups of samples might be?
So that's why we're looking at the third chapter to see how that works and if we can compare within those samples. And when we figure out how RNA later impacts it, we'll have a better idea of how we can compare the two. And we're not comparing Mobile Bay to Perdido Bay either. We're looking specifically at the environmental variation with it. Mobile Bay and Perdido Bay just as helpful with that because they both have changing environments, and Mobile Bay changes more than Perdido Bay does, and they have different freshwater discharge that can potentially be impacting it.
I think we're going to switch gears and invite our next speaker up.
I can't hear you guys.
Oh, great. I'm so glad we can hear.
Oh, I can hear you now, but it's really light.
Okay. All right, the microphone is on. Make sure you use the microphone.
Yeah.
Okay. Okay. All right. Let's go and get you loaded. It was just 1 minute for the online audience. Where is
The virus?
Yeah, go to the
All the way down the hall.
A little less. Like a floating head.
It's the reverse.
Real science.
Okay. All right. So, Brian, you're on the screen. Can you hear us?
Yes, I can.
Ready for your introduction, and when he's done, just go ahead and drag this back over. Okay. All right.
So, hi everyone. I'm going to be introducing Hari today, and I would destroy his last name. I apologize, Hari. My pronunciation is not great of your last name at this point. Hari has been here for about 3 semesters now. I guess you're finishing your 3rd semester. And he is working on a broader project, a no restore project, looking at sort of physics to fish is sort of how we sort of often talk about the project. And he's going to be focused on understanding the physics part of the project. And his work is going to be focused on Mobile Bay using some numerical simulations that we're producing for this project. And so I will turn that over to Hari.
Yeah, thank you, Brian. Yeah, still there. Okay. So, hi everyone. Let's discuss my prospectus first. So, my name is Harikrishnan Sreeshylam. That's a pronunciation. The prospectus is entitled "The Dynamics of a Strongly Stratified Channel-shoal Estuary." Is it? Yeah. Yeah. Okay. So estuaries are coastal bodies of water where the salt water gets significantly diluted with the fresh water from the river. So the fresh water, which is of less density, flows out of the system to the surface, and which is compensated by the inflow of salt water from the ocean. So this intrusion can also contribute to the lateral circulation of the system. So in the figure, looking at the center of these black arrows, the salt water advection is highest along the center, which will bring more saline water to the center in the deeper region. So the salinity will be higher in this region, which makes more dense water there, and compared to the 2 shoals. So this will make a strong lateral circulation along the bottom, like these arrows along the bottom. And this will be compensated by an outflow along the surface. So it will make a counter-rotating circulation cell as noted by these arrows. So together, this estuarine circulation can contribute to or can affect the distribution of waterborne materials, transport of sediments and water waste, and circulation of the nutrients and oxygen. So in that way, the physics can affect the biogeochemical process of the system. In addition to this, the relation between the circulation or the dynamics with the temperature is least explored in or is often neglected in studying the dynamics of the system, but it plays a role in defining the biogeochemical process of the system. And all these conditions can change by the impact of forcing such as river discharge, tides, wind, evaporation or rainfall, vertical mixing, and stratification. But the strength may change with respect to the bathymetry or the geomorphology of the system. So that motivates to individually diagnose each estuary or with varying bathymetry. So based on that, the first 3 chapters of my dissertation will look into the influence of a distinct channel-shoal estuary on its dynamics, and the 4th chapter will look into methods for improving the model temperature estimations in that same system. So Mobile Bay, as you know, it's a channel-shoal estuary,
The channel is along its length, which means it's 40-meter depth on an average, with two shoals of 3 meters on both sides. This is the channel. This is the bathymetry plot actually. So the system is river dominated, seasonally varying river discharge, and during summer, the system is highly stratified along with highest residence time that can create hypoxic bottom water along the center, especially in the channel. And because of this laterally varying bathymetry, the system has highest salinity gradient laterally, which is estimated as 10 PSU between the channel and the shoal. So this can also contribute to lateral density-driven circulation. So this is sort of what's happening in Mobile Bay. On the left-hand side, this is surface salinity, which shows the yellow color shows that the fresh water goes out of the system through the surface, and here the blue color shows the saline water that include into the system through the bottom along the channel. During summer, this salt water can intrude all along the channel and can reach far north, like near to the city of Mobile. And this is represented by the red color. And here the blue represent the freshwater outflow. So most of the previous studies have used hydrographic data like CTD data for defining the dynamics of the system. But that was just based on pointed data, like pointed observation data points actually, but that couldn't address the bay-wide dynamics of the system. So that can only be simulated or estimated using a hydrodynamic model. So even the Mobile Bay is very shallow, it's highly stratified. So only a 3-dimensional model with different vertical layers can define what's happening in Mobile Bay. So I'm still using the regional ocean modeling system with 16 vertical layers. So that's enough to represent the vertically seasonally varying trend in the system, along with forcing parameters such as climate, river runoff, open water conditions such as temperature, salinity, and the tides. And the model will simulate the bay-wide temperature, salinity, the currents, and the sea surface elevation, This is a model grid, model domain that include the delta and part of the shelf as well. So the first chapter will look into the influence of lateral bathymetry in the dynamics of this strongly stratified microtidal channel-shoal estuary. So the across-estuary variation in bathymetry can create lateral variability in the density, as I mentioned in the first slide, which means high saline water in the center can contribute to the flow along the bottom to the shoal, which is compensated by the opposite flow, making the counter-rotating circulation cells. This mechanism can contribute to the exchange of momentum over salt between the shore and the deeper region. And also in turn, it will contribute to the overall exchange flow. But the trend may change with forcing parameters such as river discharge or tides. So in Mobile Bay, being the system is laterally varying, this is a system with a laterally varying depth. The central region will have high salinity. This is just a conceptual image, like showing the high saline water along the center with fresh water on the two shoals. So this can induce strong lateral circulation, which make the counter-rotating circulation cell, which is shown by these red arrows. The water goes like this way first and making the counter-rotating circulation cell. And the trend changes with, or the strength changes with river discharge and tides. Some of the previous studies have defined this mechanism, but didn't look much deeper into the contribution of this lateral dynamics. So the objective of the study is to identify the influence of tidal asymmetry and river discharge on the lateral density gradient in driving this baroclinic circulation, and to address the contribution from lateral baroclinic circulation to the overall estuarine exchange flow. So looking into the river discharge impact of the system, during the high discharge event, the system will be highly mixed except the deep ship channel. It's clear from this figure, which shows the depth average salinity. It's highest along the channel, and all other parts are still mixed. So in this case, the lateral density will be very weak, or that can induce very weak lateral current, or maybe even zero. On the other case, during the low discharge event, the system will be highly stratified with high saline water along the center, and that can induce strong lateral current from the center to the shore, which is also clear from this figure, which is more stratified water column. And this trend may change with respect to the impact by tides. During the phase of the flood phase, the channel will have, or the central region will have high saline water compared to the two shores. And this case, the circulation or the counter-rotating circulation cell is shown by these arrows that converges at the center, the surface, and that can transport momentum downward. And it will enhance the bottom inflow of saline water. On the other case, the two shoals will have the saline water during F phase, and this will transfer momentum upward from the bottom. The convergence occurs at the bottom. That can transfer momentum upward to enhance the surface outflow freshwater. So in Mobile Bay, the river discharge impact is high compared to the tidal forcing. So the model will be simulated for two cases. In the first case, for a high discharge event, to know this trend, and then the second case for a low discharge event, just high discharge case for this one and low discharge for this one. Keeping the other parameters like tides and climate forcing such as wind the same. And the model will be simulated for 2019 year, because that was the year when the river discharge extremas was very high, with highest high and lowest low. In the second chapter, I will look into the effect of the lateral wind dynamics. How the wind can influence, the lateral wind can influence the lateral circulation. So as I mentioned in the last chapter, the stratification or high saline water in the central region can enhance the circulation. So as we know that the wind favors the decrease in stratification. So in that way, the strong lateral wind can influence the lateral circulation system. For what's happening in this case is that for shallow depth, the current flow will be in the direction of wind, and for the deeper region, it will be in the opposite direction. So in that way, strong wind can affect the lateral circulation, that counter-rotating circulation cell. In the case of Mobile Bay, as I mentioned, central region will have high saline water or stratified water during the summer season especially, and that can induce a strong lateral circulation. And one thing that need to consider here is that during the high stratification time, especially the summer, the bottom region may go hypoxic. In that case, when the wind, considering the weak wind, that may not be able to break down the stratification, but it can push the hypoxic water from the center to the shore. That causes the Jubilee event. And with the strong wind, this system may change, the stratification may break down, and that can literally influence this counter-rotating circulation cell. So this is the case of that hypoxic Jubilee event. It showed the transfer of hypoxic water from here to the shores, making that distinct Jubilee event. So some of the previous studies have already noted that the stratification may break down with the strongest cross-estuary wind. This is a study from Jeff Coogan in 2019, and it showed, and the diagram shows the time series data from the Middle Bay Lighthouse Arco station for the summer 2016. And the red shows the salinity of the bottom and surface salinity by the blue. And the difference means the system stratification. It shows it's a scale for the stratification. And here the red shows the cross-estuary wind strength, and this one shows the river discharge. So when the river discharge was very low in the summer, the strong cross-estuary wind can break down this stratification, making the water column more mixed. And this in turn will affect the lateral circulation system as well. So once the wind got relaxed, system came back to its stratification level. So which means that when the stratification is relatively weak by the action of strong estuary wind, the lateral circulation will be entirely influenced by this cross-estuary wind-driven advection. So to capture that trend or capture the effect of wind, the model will be simulated for two cases. In the simulation one, the model will be simulated for considering the wind like cross-estuary wind, both eastern and western wind. And the second case is for without wind. So comparing these two simulations can give an idea about how the lateral wind can impact the system. And the objective of the study is to characterize the role of this cross-estuary wind in altering the lateral dynamics and to describe the impact of this lateral wind effect in modifying the bioplanic circulation of the system. In the third chapter, I will look into the impact of anthropogenic modifications such as dredging and such as modification to or how those modifications can impact the estuarine properties like circulation, residence time, and stratification in this distinct channel-shoal estuary. So navigable waterways or channels are dredged or maintained to say enhance the navigation, the capacity of navigation. So this can alter the hydrodynamics and affect the basic process such as estuarine circulation and the salinity distribution. Also, it will affect the biogeochemical process associated with the stratification and the residence time of the system. So both the physical and biogeochemical properties of the system may affect by this anthropogenic activities. So it is important to identify how the system may behave with respect to these changes. So in the case of Mobile Bay, it's currently the dredging is currently going on by these new dimensions. The depth is almost about is expected to be deepening by 2 meters and the width by almost 40 to 50 meters. And it is expected to be completed by year 2025. So in the system, what's currently going on right now is because of this channel-shoal geometry, the central region will have high salinity, and that can induce strong lateral circulation, especially based on the salinity in the channel. And during summer, when the discharge was very low, the intrusion will be high, and the salt water may reach up to near to the city of Mobile. And also during the summer, system will be highly stratified with the highest residence time, and that can drive that can cause the formation of hypoxic bottom water, especially along the deeper channel. So definitely both the physical and biogeochemical properties of the system may affect by any changes that made to the channel. So checking the residence time in detail, this shows the vertical difference in water age. So water age and residence time both are time scale of physical transport processes. And during the dry season, the red color shows the highest residence time over the water age, and it's highest along the deeper channel along the northern side actually. During the winter, during the wet condition, the system has the highest residence time only along the channel and somewhere in the Bonesecor Bay region. So it concludes that largest vertical difference in water age are in the deeper channel for both seasons. And it's associated with the highest stratification level in the channel. So during wet season, the system is highly mixed except the deep ship channel. And during the dry season, the entire column will be stratified with strong stratification in the deeper channel. So in both cases, in both conditions, the deep channel has the highest stratification, or the stratification. And the circulation, the salt water intrudes into the system through the channel. And during the dry season, it can intrude very strongly and can reach far up to the north. And during the wet season, there is no such intrusion, but there is still intrusion through the channel, but it's not that strong enough to reach far north. So any modification to the channel will affect, like the modification of the channel will reduce the friction, bottom friction, and this may allow more salt water to intrude in the system. By widening the system, it can accommodate more salt water, and it will enhance the lateral circulation from center to the shores like this. So all three conditions are considered to the channel. So modification to the channel will definitely impact the system. So the objective of this chapter is to investigate how the channel deepening and widening can affect the existing longitudinal and lateral circulation pattern and the overall residence time and the vertical stratification. So in order to simulate this variation or simulate this mechanism, two simulations should be done. One for the present day bathymetry condition to know how the system works presently, and one for the proposed future bathymetry data. And two conditions will be simulated for the same other forcing parameters such as tides, climate forcing, and river discharge. And for getting the residence time, a method of Lagrangian particle tracking will be used that helps to track down the particle that placed virtually on the model domain. So till now, I have discussed the dynamics based on the salinity structure. 1 second. So the temperature is often neglected while studying the dynamics of the system. So, but the temperature plays a role in controlling the biogeochemical process of the system, and approximate or precise say estimation of temperature is essential for modeling the physical as well as biological and chemical processes in the system. So the solar attenuation changes with the clarity of water column, as you know that. And so it means that the water clarity defines the vertical temperature profile of the ocean. So Gerloff in 1976 defined 10 different water types based on the clarity of water, and later Paulson and Simpson defined a double exponential function to define the propagation of this solar light, solar radiation in the 10 different water types. So these two mechanisms, these two principles are used as attenuation parameterization in the ocean models such as ROMS. So this is the double exponential function that I mentioned. It uses coefficients from Gerloff's water type. These coefficients are different for different water types, ranging from the first one to the last one, which is the 10th one. Actually, there is 10 water types, but the 10th number is known as type 9. So this is the diagram that shows the attenuation for different water types plotted using this equation. And this ROMS model can support these spatially varying water types. So in my studies, I use the water type 9 because the water is too turbid in the system. And I simulated the model using this attenuation equation with the coefficients of water type 9. And I simulated the model for year 2019, which is shown by, the temperature is shown by this blue line. It's clear from the figure that it's unable to retrieve the seasonal changes in temperature, especially in the summer. But summer is the time when physical and biogeochemical processes are enriched in the system. So an accurate estimation of temperature is important in defining the processes. So the objective of the study is to generate a method for improving the model temperature estimations on the system and to validate the results of different methods with the in situ or ARCO stations to choose the precise estimation of temperature so that we can define the temperature related process in the system. So in the first method, I consider the concept that mobile bay is in the mid-latitude region. So the heat flux between the surface and the atmosphere plays a major role in controlling the system temperature. So what's happening actually is that the ocean surface or the bay surface is heated up by solar radiation and cooled down by the non-solar heat flux such as evaporation. So I included both evaporation and solar radiation by coupling the ocean and atmosphere, existing ocean and atmospheric model. And that coupled equation will be implemented into this existing model to validate the temperature with ARCO stations temperature. And the second model, in second method, I derived a single exponential function instead of that double exponential function I mentioned in the previous slides. So that's this one is based on the PAR data collected using different CTD cast along the bay and somewhere in the shelf. So this is the trend. The double exponential, instead of the double exponential function, the single exponential function showed a perfect fit with the light attenuation in the system. So attenuation coefficients are defined using this relation, this single exponential relation, and those coefficients are compared with the surface salinity for this CTD station, those white dots, and a relation showing by this y equal to ax raised to b. That's a new relation based on the surface salinity. Here the surface salinity is the variable, and this will be implemented into the existing model in place of the conventionally used double exponential function. And both cases will be compared with or validated with ARCO station temperature to define the perfect match or the perfect temperature predictions that can, so that we can define associated chemical or biological process in the system. So that's all about the prospectus, and I would like to thank the people in my group, Dr. Brian, Dr. Lateral, Dr. Silong, and Dr. Lisa from the NC State University, and tech support for the field data, and Steve Dykstra and Jeff Dugan for some previous data of the mobile bay, and Nova for funding the project. So thank you. Questions?
One of your last slides, you talked about the coupling of the ocean and atmospheric models. The cooling aspect was just evaporation. Would there also be a major component of cooling based on mixing?
Yeah, there is mixing, but we're not considering.
Sorry, the question into the microphone.
So he asked, is mixing plays any role in controlling the cooling, right?
Right.
Okay. So mixing is there, but we are not considering the part of mixing. It will be complicated. Yeah. Yeah.
Can you go back to slide 10?
10. This one?
Yep,
Okay.
that one. You see that point all the way up in the bay on that one. Keep on going up. That one right there. That
This
shows the higher salinity waters, and you see it in the same time. In either time period. Do you know what's going on there? Is that Arlington Channel or something? Because that.
Here?
Yeah, that higher salinity pulse there, the fishing guides and the crabbers all use that area extensively, no matter what the season is, because it always seems to have good catches of fish and crabs. And I was just wondering what's causing that higher salinity at that point.
That's in the channel.
It's in the channel.
Can you repeat the question into the microphone?
So he asked, what's the reason for this high saline water in this region? So I think this is because of the channel, right, Joe? Yeah.
But it seems even higher there than the rest of the channel.
Well, so there is.
It's because of the residents. So I think it's because the water stays there for a long time. I think so. Yeah.
You know, one thing we got out of working up in that area in the Delta is also in that same spot he's talking about. Oxygen tended to be lower in the water column for some reason, in Polecat Bay too.
And that leads to kind of my second question, which is slide 19, the age of the water.
Yeah.
What does that vertical difference to in water age? Is it the bottom that's old?
Yeah, from the surface and the bottom.
Okay.
Yeah.
And why is that off that?
Here?
Yeah. What's causing that water to stay there for so long?
I think this is because of it's dry season, so the river discharge impact may be very low. So only salt water can push the water, so it will stay there. It's because of the river discharge, maybe.
Yeah, John, I wouldn't think of it as a staying there. It's sort of like if you have a particle released there, how long does it take to sort of leave the estuary? And so stuff that's there takes longer to get out of the estuary than other places. And so stuff on the bottom, knowing the residual circulation in an estuary, water comes in from the bottom, has to recirculate to the top and then come out. There's a big vertical difference there because the bottom water just takes longer to get to the surface and then get evected out of the system. And so I wouldn't think of it as water stays there. It just takes longer to cycle through the system if you're originating from that part of the bay.
Okay, well, obviously I'm not as smart as a physical oceanographer, but I'm not sure how that is different than staying there. But I digress. Like I said, if I was smarter, I would have been a biologist.
Yep.
Would you go back to slide 10? You see that one spot up there? If you were to shoot a dolphin, that would its skin bacteria look different than somewhere else in the bay since the water stays there longer?
Don't worry about that. Unfortunately, we gave him cake.
You're stuck with me now, Powers.
So into all of these models, you have the realistic tidal forcing and river discharge. What are you basing that realistic clarification on?
The real data.
From what years?
2019, the year 2019. So
Just 2019?
yeah, so all the simulations will be on the year 2019.
Okay, and then for the realistic climate forcing, is that how you're going to see how those things are essentially going to change over the future, right? Because is that how you factor in change into how you're going to look at this? You understand what I'm saying?
No.
I don't know. So you have these realistic ones that are based on this 1 year in 2019, and then you have this climate forcing that you're putting in, and that should hypothetically be how these things are going to change in the future, right?
Yeah.
Okay. That was my
Got it? Yeah. Carter?
Yeah, so you had this one slide where you looked at how strong winds would break down the stratification. Roughly, how long does it take for the stratification to come back and start circulating again? Because that one graphic showed us it did re-stratify a bit, looked like it was less stratified than it was before the high winds. So how long does it take to go back to normal?
To re-stratify the column?
Yeah.
So like in this
After
figure?
high winds, yeah, like that.
So from this study, they showed it got re-stratified in a couple of days, I think. In a couple of days.
Well, because what I'm curious about is that the stratification seems to still be less after the winds than it was before.
Yeah.
Even after.
So it's still stratified, but the strength is not same as what it was before. Yeah.
Oftentimes, see what happens is it will mix, but it'll also just push the isopycnals up on the shoals. So they'll get vertical, and it looks like it's mixed, and then when it relaxes, it just flows back out. So you can get stratification, re-stratification often within hours. Sometimes it hasn't.
Yeah, I mean, the way you can think about this is, once the bay mixes, right? So you've now taken ocean water and fresh water and basically turned into one thing, right? So now it's this brackish water. And until that whole bay clears itself of that brackish water, there's no way to get sort of ocean water back on top of fresh water, right? So it takes longer to re-stratify to the level before the mixing event because you have to basically clear the whole system of that brackish water so that you can get salt water sitting on top of fresh water, right? And so it takes longer to re-stratify, the immediate, there's immediate re-stratification that's not as high as what was pre-storm or pre-mixing event, because you have to clear all the water that just got stirred up in order to get that, really dense ocean water back into the system.
That makes sense. Thank you.
Yep.
So You're talking about the effects of the deepened ship channel.
Mhm.
Could lead to more salt water up onto the shoal.
Yeah.
Locally, that has interest because of the Jubilee events on the eastern shore. So if a deepened ship channel would possibly create more of that, could you extrapolate backwards and look at before the ship channel was dredged?
I mean, before all the dredging, like,
200 years ago.
okay,
To see if there's record of Jubilees happening far less frequently than the past 100 years.
that's possible. Yeah, that's cool.
Yeah, so.
The other thing I would add is that I think one of the questions we're trying to answer is how much of that channel water contributes to Jubilee events. It's not clear, to me at least, that hypoxic water from the ship channel is spilling out and causing the Jubilees. It could just as easily be locally generated, so the flank going hypoxic and that water just getting moved closer to shore. I think one of the goals of this work is to better understand the connection between the ship channel and the flanks. We don't have a great grasp of that in the system. And so understanding that communication between that deep ship channel and the flanks is one of these questions we're hoping to get to the bottom of.